Hello everyone, my name is Danielle. I'm a junior student at Texas A&M University, San Antonio. And today I would like to further discuss Typhloidea cave shrimp and specifically how they respond to salinity and environmental change and just some trends that we have seen in our transcriptomic data so far. Before I begin, I just want to thank my mentor, Dr. Elizabeth Borda, and as well as our collaborators and co-authors on this project. So to begin, first I will just briefly discuss Typhloidea cave shrimp and their different species salinity preferences. Then I will go over our transcriptomic pipeline and methods, and afterwards I will take a look at the candidate genes related to osmoregulation and oxidative stress specifically. And lastly, I will just summarize our results so far and discuss possible next steps for our work. So, Typhloidea is a part of the Anchialine family, which is composed of about 450 species that are predominantly freshwater. But within the genus Typhloidea, these species of shrimp are specifically anchialine adapted and are found within marine, brackish, and freshwater environments. And then looking even closer into the Yucatan Peninsula, we find 5 species there, broadly distributed, but only one of these species has been identified as Euryhaline, and that means they are existing within both the brackish, marine, and freshwater environments. The other 4 species, which were previously discussed, are only found within the freshwater environments. So if you look at this next slide here, you can kind of see a little bit more of the distribution. The blue shrimp represent Zilamensis, and so they are collected both in the marine and freshwater environments, and then near the halocline brackish mixing zone as well. The other 4 species all above the freshwater, and so that is that group. And then, so our research objectives for this project, first we wanted to functionally characterize the transcriptome of the Yucatan Typhloidea species, specifically focusing on that one Euryhaline Typhloidea zilamensis species. From there, we would then identify genes associated with osmoregulation, and as well as oxidative stress and respiration, and then to get a better understanding of the tolerance and preferences, or saline preferences of the species. And then lastly, we just wanted to evaluate differential gene expression in response to that salinity change within the species. So looking at our transcriptomic pipeline here, we begin by collecting the shrimp from their anchialine native environments, and then what we did is we exposed one group of Zilamensis shrimp to fresh water, and then another one to brackish water for 15 minutes, and then we have the control group as just the Zilamensis species that were collected from the native marine water. And from there, we were able to isolate the RNA and reverse transcribe it to get cDNA for Illumina high-seq sequencing to create a cDNA library database, which we could then use for further analysis. So to begin, we had just our raw sequences, which were then filtered through a FastQ pre-processing step. To do that, we ended up eliminating some low-quality reads and filtered out the adapters as well. From there, we took those clean, filtered FastQ files and were able to de novo assembly those using the Trinity Sequence Assembler software. The sequencing data from the individuals treated, the 3 individuals treated within the same brackish water or freshwater groups were concatenated to create and be treated as one whole specimen for the following steps. So we had those groups which were then de novo assembled and put through a protein coding transcript step. which then identified areas in the transcriptome which could be seen as potentially protein coding. And after that, we could continue on with the functional annotation step, which then allowed us to take the transcripts that we had and assign the functions and kind of go categories, the gene categories of those transcripts. From there, we could then do candidate gene screening, which allowed us to look for transcripts specifically related to osmoregulation, and then oxidative stress and a few other categories as well. And from that step then, we could continue on with the differential gene expression and the statistical Fisher's exact test. So just to kind of give an overview of our data, for the freshwater, we started out with about 350,000 transcripts. That was after the de novo assembly, and then the protein coding identification step, we had 105,000 contigs with an N50 number of 871, so about at least 50% of those contigs had at least 871 base pairs. And then from that list, 62,171 were actually annotated during the functional annotation step. And then you can see the results for the brackish samples there as well, transcripts and contigs. And then with our completeness, we had a 92.6% reported completeness for the freshwater and 90.4% reported completeness for the brackish. So moving on to the candidate genes for osmoregulation and stress, we primarily worked off of a previous study that looks at candidate genes within Macrobrachium, which is the freshwater shrimp found in Australia. And so using this list of approximately 40 genes associated with osmoregulation, we were able to screen through our annotations and identify ones related with this list. Some of these genes were also looked at in another study that was done with Halocaridina, which is the shrimp here in Hawaii. And so just as some examples, we had heat shock proteins, which are used for response to stress, carbonic anhydrase, zinc ion binding, and so on. So those were the kinds of, just some examples of the kinds of genes, identities we were looking for. So in another study here, you can see there was a study conducted with T. Mitchellii, Pearsi, and zillamensis. So Mitchellii and Pearsi are the freshwater adapted species, and zillamensis is the Eurohaline one. And what happened was the shrimp were subjected to physical activity in order to encourage respiration. And from there, they were able to monitor indicators of oxidative stress, anaerobic metabolism, and associated genes with the antioxidant system response. So with that, we were then able to actually use the data results that we had and compare it to this list as well, and found that a lot of our results actually matched what they found, which was an upregulation of genes associated with these responses. So you can see here in the list, we have there's the heat map, and then we have the Fisher's exact test. And in there, that gives the general categories of genes that were upregulated. So you can see like non-membrane bound organelles, ribosome, chromatin, kind of genes related to activity at the cellular membrane. And then we also looked at the differential expression, and these are both upregulated. So there was an overexpression of these genes within our test set, which was the zillamensis freshwater sample when compared to the zillamensis brackish water samples. And so these were upregulated within that group. And you can see kind of more of the examples here of peroxidase, more heat shock proteins, and glutathione, etc. And then also to report, we have our general categories again for the Fisher's exact test that are under-regulated. So we have ones related to transmembrane transport activity and oxidoreductase activity as well. And the heat map on that side shows the same as before, the upregulated freshwater compared to brackish. So what we found is that we were able to functionally characterize the transcriptome of Typhloidea zillamensis and specifically screen for those approximately 40 genes associated with osmoregulation, as well as oxidative stress and respiration. And then we were able to perform a differential expression and observe differences in response within zillamensis to the different salinity exposures. In total, there's about 10,327 total genes that were identified as either up or down regulated. 3,000 or about 4,000 of those were up regulated specifically. And so within that group, we were able to also notice that our results matched what was also done with previous experimental work as well. But then we were able to expand upon those results with our transcriptome data. And so on that note, for future work, we would like to analyze transcriptome data specifically from Typhloidea zillamensis that was collected in the marine water, so the control group, and then do a three-way comparison between the different salinity levels for zillamensis. And then going even further, what we would like to do is conduct comparative functional transcriptomics with freshwater T. pearsi and T. mitchellii related species and see any differences in expression within those groups when exposed to the different salinities, since those are indeed the ones that are freshwater adapted. And just once again, thank you to all of our collaborators. And this was also funded by NSF STEM. So thank you, and thank you for having me.
Any time for one question?
Yes.
Yeah, I have two questions, so you can answer whichever one you want. But,
sure.
What was your sample size for your differential expression? And then...
So, yeah. What's the other one? Sorry.
Sorry.
I didn't mean to cut you off.
Yeah, go ahead.
Got
Just
excited.
answer that one.
Okay. The sample size. So we had 3 samples for each of the salinity groups. So the freshwater, brackish, and then we also have marine. We just need to finish the processing of that data. Okay. And then it's the same. We also have samples from pearsi and mitchellii, and also 3 individuals. The difference was that we concatenated the 3 individuals to then be treated as one whole organism and used paired differential expression without replicas as our methods to use as one whole transcript or one whole organism. So that was kind of the difference. We started with 3, but then we kind of combined them and pooled them into one for each salinity. Any other questions? Okay.
Thank you.